Welcome children. In our last session we discussed about the spermatogenesis. Today we'll discuss oogenesis. Then we'll discuss how oogenesis is different from spermatogenesis. Or we can discuss the comparative account of oogenesis and spermatogenesis. Now this oogenesis is initiated, very important. Oogenesis, it is initiated at the embryonic stage. It is initiated at the embryonic stage when the embryo is in the mother's womb. But in the spermatogenesis starts at puberty. So this is the difference. Okay. So this process, let me first explain the process, then I will explain you how exactly in which stage the egg is released and all these things. So it starts with, if you will take the structure of a ovary, this is the structure of ovary. Then in the ovary we can see that here the blood vessels are supplied to the ovaries, and inside the ovary there is a lining of epithelial cell. Okay, this lining is called germinal epithelium. This is called germinal epithelium, which consists of epithelial cells. So this is epithelial cells. Okay, now this epithelial cell, as the process of oogenesis takes place, this epithelial cell, one more thing, let me tell you, let me remind you that the ovary is differentiated into two parts. That is the outer epithelial lining and the middle part is known as stroma. Now this stroma is differentiated into two parts. The central part is the medulla and this peripheral part is the cortex. Okay. So as the process of oogenesis takes place, we see the oogonial cell at different stages. So like the primary follicles, then we'll see the secondary follicles, tertiary follicles, like this. I will tell you what are these follicles. Okay, let me first draw it. Then it forms the Graafian follicle. So like this, this is the primary follicle, secondary follicle, then this is tertiary follicle, and then finally it forms the Graafian follicle. This is follicle. And then you will see a stage where the Graafian follicle is ruptured and the ovum is released. This is called ovulation. So this ovum is released in the stage of secondary oocyte. Okay. Then the remnant of the tissues of Graafian follicle, it forms a structure. The remaining part, it forms a corpus luteum. Okay. Now keep it in your mind, this diagram. Now let me explain the process of oogenesis. The oogenesis starts with oogonium. Okay, but spermatogenesis starts with spermatogonium. Now this oogonium, there is no division so far, so it is diploid. That means it is having 46 chromosomes. And this oogonium undergoes mitosis to form a number of oogonial cells, all of which are 2n. Okay, and this division is mitosis. Then here I am showing only one oogonial cell. This is oogonium. Okay. Now this oogonium undergoes differentiation to form the primary oocyte. This is primary oocyte. So the primary oocyte is formed simply by differentiation. So its ploidy is also 2n, means 46 chromosome. Okay. Now this primary oocyte, when a girl baby is born, it is born with this stage, primary oocyte stage. Okay, and after birth, no more. So at the time of at birth, at the time of, please note it down, time of birth, the ovary contains, each ovary, okay, each ovary contains millions of gamete mother cell. Okay, millions of gamete mother cell. Okay, and these gamete mother cell divides and it enters. Actually, the primary oocyte is present inside the primary follicle. So here what happens, the cells of the cortex are gathered here to form a structure which is known as primary follicle, inside which the primary oocyte is present. So here what is present? Primary oocyte. This is primary oocyte and the surrounding cells are follicles. So it forms the primary follicles. So these primary follicles are present at the time of birth and the primary oocyte is present inside the primary follicle. Okay. Now this primary follicle, in the primary follicle, the primary oocyte under the meiosis starts in the primary oocytes when it is inside the womb of the mother. So here what happens, in the primary oocyte, the meiosis is started but arrested at prophase 1. अभी आपको ध्यान से सुनो बच्चों, प्राइमरी oocyte, जैसे प्राइमरी स्पर्मेटोसाइट को क्या करना था, मियोसिस 1 होना था, तो मियोसिस 1 होता था. अभी प्राइमरी oocyte में भी मियोसिस 1 शुरू हो जाता है बिफोर द बर्थ ऑफ द चाइल्ड. लेकिन ये जो प्राइमरी मियोसिस 1 शुरू हुआ है वो कहाँ आके रुक जाता है, प्रोफेज 1 में आके रुक जाता है. सो ये जो प्राइमरी oocyte, जब बच्चा पैदा होता है उसके ओवरी में प्राइमरी oocyte इज प्रेजेंट इनसाइड द प्राइमरी फॉलिकल. सो प्राइमरी फॉलिकल क्या है, सो लेट मी राइट डाउन हियर, द प्राइमरी फॉलिकल्स it contains the primary oocytes surrounded by granulosa cells. ओके, सो आपको जो मैंने कॉर्टेक्स बताया, उस कॉर्टेक्स में आपका जो फॉलिकल बन रहा है, हाउ दिस फॉलिकल इज फॉर्म्ड, the follicle is formed by the combination of the primary oocyte and the granulosa cells. Okay, so a layer of granulosa cell is granulosa cell surrounds the primary oocyte, and this is called primary follicle. So let me show you with the help of diagram. So suppose this is the layer of cell, and here is this is primary oocyte, and these are granulosa cells. Okay, and together it constitutes the primary follicle. So these primary follicles gradually degenerates during the phase from birth to puberty. So very important, so at the time of birth each ovary was having millions of these primary follicles, but from birth till puberty what happens, many follicles, a large number of follicles degenerates. Okay, and at puberty only 60,000 to 80,000 primary follicles are left in each ovary. Primary follicles are left in each ovary. Very very important children. So now ये हो गया आपका primary follicle. So now primary follicle with primary oocyte is formed. So this primary oocyte is in the primary oocyte the meiosis 1 has been started, but it is arrested at prophase 1. So this is at the time of birth, and this stage continues till puberty. And at the onset of the puberty, जब puberty आती है तब क्या होता है, the primary follicle develops into secondary follicles, then tertiary follicles, it gets surrounded by more granulosa cells, and then it is called secondary follicles. Now what happens to this primary oocyte? Now what happens, this primary oocyte, so here let me write down. At puberty, so this is at embryonic stage. Okay, the oogenesis starts at embryonic stage, very very important children. Then at puberty what happens, the primary oocyte releases from the prophase 1. अभी जो प्रोफेज 1 में रुक गया था, अभी प्रोफेज 1 से ये रिलीज हो जाता है, मतलब प्रोफेज 1 से प्रोफेज 2 में वो एंटर कर लेता है. इट एंटर्स इनटू द प्रोफेज नॉट 2, प्रोफेज 1 के बाद मेटाफेज 1, टेलोफेज 1, अनाफेज 1, टेलोफेज 1 कंप्लीट कर देता है. सो व्हेन इट कंप्लीट्स, it divides into 2, but this division is unequal division. This is unequal division. So at puberty what happens? At puberty it releases from prophase 1 and completes meiosis 1. मियोसिस 1 कंप्लीट हो जाता है. तो जब मियोसिस 1 कंप्लीट हो गया तो आपका क्या हुआ, 2 सेल बन गए. सो 2 सेल्स आर फॉर्म्ड, बट दिस डिवीजन इज अनइक्वल डिवीजन. सो हियर व्हाट हैपेंस, वन ऑफ द सेल इज स्मॉल एंड अदर सेल इज बिग. ओके, सो दिस बिगर सेल इज नोन एज secondary oocyte. Okay, and this is called, this small cell is called first polar body. First polar body. Clear? Now this first polar body, it may divide or it may not divide, and it may degenerate. Okay, now what happens, this secondary oocyte is contained in the secondary follicle, then Graafian follicle. So what happens now, more granulosa cells are accumulated around the primary follicle and forms the secondary follicle. Okay, so now initially in the primary follicle only one layer of cell was there. Okay, now one more layer is formed, so it is known as secondary it is known as secondary follicle. And then this secondary follicle develops into tertiary follicle. And this tertiary follicle, how it is different from secondary, here what happens, a cavity is formed. Okay, and it contains the secondary oocyte. So these are the granulosa cells present at the periphery. Okay, and here this is the antrum. This is antrum, and here is the secondary oocyte. And what is this structure? This structure is tertiary follicle. Okay. Now this tertiary follicle develops into Graafian follicle. So in the Graafian follicle, the structure is somehow like this. So here is the secondary oocyte. And this secondary oocyte is surrounded by a layer of cell that is known as zona pellucida. Then it is again surrounded by the cells called corona radiata. Then here is the antrum, and this is the theca. Okay, so this is the structure of a Graafian follicle. Now the secondary oocyte is surrounded by the zona pellucida, the membrane, and it is surrounded by a mass of cell that is corona radiata. So the secondary oocyte, here what happens, the secondary oocyte which is formed, now this secondary oocyte, the meiosis 2 is started. So meiosis 2 का क्या क्या phases है, सब phases है, prophase 2, metaphase 2, anaphase 2, telophase 2. So here what happens, the meiosis 2 starts but arrested at metaphase 2. देखो ये स्पर्मेटोजेनेसिस से कितना कॉम्प्लिकेटेड है. देखो पहले आपका प्राइमरी oocyte तक आपका जो oogoneum डिफरेंशिएशन होकर बन गया. प्राइमरी oocyte में मियोसिस 1 शुरू हो गया, बट ये मियोसिस 1 प्रोफेज 1 में आके रुक गया. एंड इन दिस कंडीशन, इसी कंडीशन में ये ओवरी में होता है और बच्चा पैदा होता है. बच्चा जब पैदा होता है तो इस कंडीशन में जो oogenesis का इसी फेज में आपका जो एग होता है. Now after birth, these primary oocytes are contained in the primary follicles. After birth, gradually till puberty, धीरे धीरे जो प्राइमरी फॉलिकल्स है वो डीजेनरेट होने लगते हैं. एंड एट प्यूबर्टी एंड ओनली 60,000 to 80,000 प्राइमरी फॉलिकल्स आर रिमेनिंग. एंड जब प्यूबर्टी शुरू हो जाता है, जैसे ही प्यूबर्टी में पहुंचता है तो आपका जो प्रोफेज 1 में रुक गया था ये डिवीज़न, प्रोफेज 1 से रिलीज हो जाता है. मेटाफेज 1, अनाफेज 1, टेलोफेज 1 कंप्लीट कर लेता है. कंप्लीट करके ये कंप्लीट डिवीजन हो जाता है. एक छोटा सेल होता है जिसको हम बोलते हैं फर्स्ट पोलर बॉडी, और जो बड़ा सेल होता है उसको हम बोलते हैं सेकेंडरी oocyte. तो अब ये सेकेंडरी oocyte का प्लॉइडी क्या होगा, बिकॉज इट इज प्रोड्यूस्ड बाय meiosis, so its ploidy is N. Remember, its ploidy is N, so polar body is also N. But this division is unequal division, because of which one is small, very small cell, and other is larger cell. The small cell is not having cytoplasm, so it degenerates gradually. Now what happens to this secondary oocyte? This secondary oocyte is now contained initially in the tertiary follicle, as I have showed you here. So पहले tertiary follicle में होता है, then it is contained in the Graafian follicle. This is Graafian follicle. तो ये जब Graafian follicle में होता है तब ये मेटाफेज 2 तक आके रुक जाता है. दैट मीन्स मियोसिस शुरू हो गया, मियोसिस 2 शुरू हो गया, बट मेटाफेज 2 में रुक गया. नाउ इन दिस स्टेज the secondary oocyte is released from the ovary during ovulation. ठीक है, अभी मैं आपको ऊपर वाले डायग्राम में ले जाती हूँ. देखो यहाँ पर हमने आपको डायग्राम दिखाया था, ये था आपका ग्राफियन फॉलिकल जिसमें आपका सेकेंडरी oocyte था. नाउ दिस ग्राफियन फॉलिकल कम्स टू द सरफेस एंड इट रिलीजेस द सेकेंडरी oocyte. ठीक है, लेकिन ये जो सेकेंडरी oocyte आप पुराने बुक में देखोगे वहाँ पर ओवम दिया है, दैट इज रॉन्ग. सो द अभी न्यू बुक में आप देखो दिस इज सेकेंडरी oocyte, दिस इज ओनली द करेक्ट आंसर. सो सेकेंडरी oocyte स्टेज में एग रिलीज होता है ओवरी से एंड दिस प्रोसेस इज कॉल्ड ओवुलेशन. दिस इज कॉल्ड ओवुलेशन. सो द रिलीज ऑफ एग from the ovary is called ovulation. In which stage it is released? It is released in the secondary oocyte stage, where it has started the meiosis 2 but ceased at metaphase 2. Metaphase 2 में रुक गया है. Now when this metaphase 2 will complete? Now when the sperm penetrates the ovum. Ovum यानी secondary oocyte means ovum. It releases from metaphase 2 and meiosis 2 is completed. तो ये कब होता है? एट द टाइम ऑफ फर्टिलाइजेशन. जब आपका ये है सेकेंडरी oocyte जब रिलीज होता है, इसके चारों तरफ क्या होता है? द जोना पेलुसिडा इज देयर. जोना पेलुसिडा. ओके, जोना पेलुसिडा के साथ सेकेंडरी oocyte. दिस इज सेकेंडरी oocyte रिलीज होता है. और जब इसके पास स्पर्म आता है एंड द स्पर्म पेनिट्रेट्स इनटू द एग, जब एग को पेनिट्रेट करता है, देन there is a change in the cytoplasm which stimulates the nucleus to complete the meiosis 2. meiosis 2 complete करने के लिए उसको stimulate करता है. और जो मेटाफेज 2 में रुक गया था, अभी मेटाफेज 2 से रिलीज होके ये meiosis 2 complete कर देता है. and now the secondary oocyte meiosis 2 is completed and it forms a second polar body and finally the ovum. Okay, so this is the process of oogenesis. Now let me summarize once again. देखो, oogenesis starts with the oogonium and it starts at the embryonic stage. Okay, the oogonium divides mitotically to form continuously. It go on dividing and it forms a large number of millions of oogonial cells, or you can say gamete mother cell. Now this gamete mother cell undergoes differentiation. It happens in the womb of the mother only. Differentiation, it develops into primary oocyte. Now this primary oocyte gets surrounded by the granulosa cells present in the cortex of ovary and it is called primary follicles. The structure is called primary follicle. The primary oocyte enters into meiosis 1. meiosis 1 शुरू हो जाता है when ये oocyte embryonic stage में होता है. meiosis 1 started but arrested at prophase 1. prophase 1 में आके रुक गया. तो जब बच्चा पैदा होता है, ए बेबी गर्ल बेबी इज born, it is born with millions of primary follicles in each ovary. but जैसे जैसे ये प्यूबर्टी तक जाती है, प्यूबर्टी तक only 60,000 to 80,000 प्राइमरी फॉलिकल्स रिमेन्स. बाकी धीरे धीरे डीजेनरेट हो जाता है. जैसे ही प्यूबर्टी में एंटर करता है तो प्रोफेज 1 से रिलीज होकर मेयोसिस 1 कंप्लीट हो जाता है. कंप्लीट होके ये अनइक्वल डिवीजन होके एक छोटा सा सेल बनाता है, एक बड़ा सेल बनाता है. द स्मॉल सेल इज नोन एज फर्स्ट पोलर बॉडी एंड द लार्ज सेल इज नोन एज सेकेंडरी oocyte. ये सेकेंडरी oocyte में भी मियोसिस 2 स्टार्ट हो जाता है, बट आके रुक जाता है कहाँ? मेटाफेज 2 में रुक जाता है. मेटाफेज 2 में रुक जाता है एंड इसी स्टेज में ओवरी से रिलीज होता है सेकेंडरी oocyte which is surrounded by the zona pellucida membrane. It is surrounded by a membrane that is called zona pellucida. तो आप देखो, आप यहाँ पर एक सवाल आप मुझे पूछ सकते हो कि टर्शियरी फॉलिकल एंड ग्राफियन फॉलिकल में डिफरेंस क्या है. टर्शियरी फॉलिकल में देयर इज नो जोना पेलुसिडा, बट ग्राफियन फॉलिकल में सेकेंडरी oocyte के चारों तरफ एक लेयर बना है, एक मेम्ब्रेन बना है विच इज नोन एज जोना पेलुसिडा, जो कि यहाँ पर नहीं है. ठीक है, सो दिस इज द डिफरेंस. सो अभी क्या होता है, जोना पेलुसिडा के साथ सेकेंडरी oocyte रिलीज हो गया. नाउ दिस सेकेंडरी oocyte रीचेस द अम्पुला, ओवरी का ओविडक्ट का अम्पुला तक पहुंचा. ओके, लेट मी ड्रॉ द डायग्राम वंस अगेन. इसलिए मैं बोल रही हूँ आपको सब कुछ पढ़ के रेडी होना है. तो यहाँ से आपका ओवरी से रिलीज हुआ एंड द एग हैज बीन रीच्ड हियर. ये एक कौन सा स्टेज में है? सेकेंडरी oocyte. ठीक है. एंड व्हेन द स्पर्म कम्स टू द ampulla, दिस इज ampulla. अगर स्पर्म आता है एंड स्पर्म इसको पेनिट्रेट करता है, the cytoplasm gets disturbed and it stimulates the nucleus of the secondary oocyte to complete the meiosis 2. जो अरेस्ट हो गया था मेटाफेज 2 में, वहाँ से रिलीज हो जाता है, meiosis 2 कंप्लीट हो जाता है. अगेन it is an unequal division. One is smaller, one is larger. So this is called second polar body and this is the ovum. Now this ovum is actually the fertilized ovum. This is fertilized ovum. So this is how the oogenesis takes place, and also I have shown you the diagram of ovary. Now if after the release of the ovum, if the fertilization takes place, then it will lead to pregnancy. The embryonic development will take place. Okay. After releasing the oocyte, what happens to the remaining tissue? The remaining tissue forms it becomes glandular and it is known as corpus luteum, which secretes the hormone progesterone. It secretes progesterone hormone. And if the fertilization will not take place, because fertilization is a matter of chance only, so if the fertilization will not take place, then gradually this corpus luteum will also degenerate. Okay, so this is all about oogenesis. So please go through spermatogenesis and oogenesis, and for you the homework is compare and contrast between spermatogenesis and oogenesis. So oogenesis is much complicated whereas spermatogenesis is very simple. तो ये दोनों को आपको कंपेयर करना है और कंट्रास्ट भी करना है. So write down everything point wise. Okay. So for today this much only. In our next class we'll discuss menstrual cycle. So till then, bye bye. Thank you.